In 1928, Alexander Fleming discovered that a fungus contaminating his petri dishes had released a powerful antibiotic that killed the surrounding bacteria. He named this antibiotic penicillin. Nearly 2 decades later, after years of intense cooperation between scientists, manufacturers, and government agencies, penicillin was made broadly available. In 1945, using X-ray crystallography, Dorothy Hodgkin determined the chemical structure of penicillin, a discovery that fueled the quest to synthesize the drug. In 1952, a form of penicillin suitable for oral use, called Penicillin V, was developed. And in 1957, Penicillin V was produced synthetically for the first time, laying the foundation for the development and synthesis of new penicillin antibiotics. Penicillin is effective for treating bacterial infections because it disrupts an essential process in the bacterial life cycle, the creation of the bacterial cell wall. The cell wall in most bacteria consists of a lipid bilayer membrane and a mesh-like peptidoglycan layer. Gram-positive bacteria build a thick peptidoglycan sheath around a single membrane, while gram-negative bacteria typically build a thin layer of peptidoglycan between two membranes. Water constantly enters bacterial cells by osmosis, building up pressure on the cell membrane. Peptidoglycan allows the cell to resist this pressure by providing structural support for the membrane. Penicillin prevents bacteria from building their peptidoglycan layer, causing the bacteria to burst under pressure. Let's take a closer look at how penicillin acts at the molecular level. Peptidoglycan is made up of small building blocks, each composed of 2 sugars connected to a short chain of amino acids with a peptide bridge extending to the side. These sugars are assembled into chains, which are then cross-linked via the peptide bridges to form a tough peptidoglycan matrix. The enzyme D-alanine carboxypeptidase transpeptidase, also known as penicillin binding protein, assists with peptidoglycan matrix assembly by creating the cross-links between the chains. Penicillin antibiotics block this enzyme by making a direct bond to a key serine amino acid in its active site. The active portion of penicillin is a beta-lactam ring. It is chemically reactive and opens up to form a bond to the active site serine. This inactivates the enzyme and prevents proper formation of the peptidoglycan matrix. The beta-lactam ring is found in both natural and synthetic antibiotics that are structurally similar to penicillin. Beta-lactam antibiotics are effective against many types of bacterial infections and have helped to save countless human lives. However, bacterial communities are living things, and like any other living thing, they adapt to survive. To protect themselves from antibiotics, bacteria developed resistance mechanisms. For example, MRSA, a strain of Staphylococcus aureus, expresses penicillin binding protein 2A, which has an altered active site that doesn't bind beta-lactam antibiotics. Bacteria can also express special beta-lactamase enzymes, which bind to beta-lactam antibiotics and break the essential beta-lactam ring, making the antibiotic ineffective. Medical scientists work quickly to develop new drugs to fight resistance. Some of these drugs block beta-lactamases directly, leaving the antibiotic free to inhibit penicillin binding proteins. Other new beta-lactam drugs are either not recognized or not broken down by some beta-lactamases. Unfortunately, in the face of overuse and misuse of antibiotics, bacteria continue to develop ways to resist the action of new drugs. Many genes that are crucial in antibiotic resistance are encoded on small circular pieces of DNA called plasmids. These plasmids can be passed from one population of bacteria to another, and then from generation to generation. One such particularly dangerous gene is NDM1. Bacteria possessing this gene can build the New Delhi metallo-beta-lactamase enzyme. These types of enzymes can break down almost all known beta-lactam drugs, posing a major global health threat. Today, less than 100 years since Fleming's discovery, we are in the middle of an arms race with pathogenic bacteria. Using our growing knowledge of the structural biology of bacterial enzymes, the medical community is looking for new ways to target the weak points of bacteria.